We'll go over the carbohydrates and lipids in this lecture. So carbohydrates have two functions. They serve as an energy source and they're also structural. So for example, cellulose makes up the cell walls in algae, and also chitin makes up the cell walls in fungi. So the structure of the carbohydrates, they all have a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. So this is how they got the name carbohydrate, because carbo is referring to carbon and hydrate is referring to water. So you see the 1 to 2 to 1 ratio of the carbon to hydrogen to oxygen. So that's what you always see with the carbohydrates. So that's important to remember. So we've got a couple of different types of carbohydrates. We have the monosaccharides, sometimes also called simple sugars. And remember, mono means one. So these are just one sugar. And these, remember, are the monomers or the building blocks of the polysaccharides. So that's important to remember. So these would be, for example, glucose and fructose. These would be examples of monosaccharides. So the disaccharides, di meaning two, you would have two monosaccharides joined together by covalent bonds. And then polysaccharides, poly meaning many, you've got three or more monosaccharides joined together by covalent bonds. And the polysaccharides, this is the polymer. Okay, and just here's how a disaccharide would be formed from two monosaccharides. Here we have glucose and fructose. We would just do a dehydration synthesis reaction. We'd squeeze the water molecule out, and with the help of an enzyme, and join them together by a covalent bond, and this would form table sugar or sucrose. And then we could also do the reverse reaction and break the sucrose apart into the two monosaccharides. We could just do a hydrolysis reaction, again with the help of an enzyme. And then our polysaccharides, this is our polymer. So we've got three or more monosaccharides joined together by covalent bonds. And so these are going to be used for energy storage. So for example, starch and glycogen would be energy storage forms of polysaccharides. And they can also be used for structure. So cellulose, which makes up the cell wall of algae, and it's also part of biofilms. And then chitin makes up the cell wall of fungi. So those would all be structural polysaccharides. All right, our next organic molecule would be lipids. And lipids are a very diverse group. And why they are all grouped together is because they're nonpolar, and that makes them all hydrophobic. So they do not like to mix with water because they've got those nonpolar covalent bonds. They're sharing electrons equally, so there's not going to be any partial positive or negative charges anywhere. So nothing to attract the polar water molecules at all. So they're hydrophobic and do not like to mix with water. Okay, the other thing I would know is the functions of lipids, what they're used for. so they can definitely be used as an energy source. And they're also used for structure. So we do find lipids as part of the plasma membrane, so like phospholipids. And they're also found as part of the cell wall. So if you think about the gram-negative bacteria with their outer membrane, those lipopolysaccharides, that would be a good example there. So we're just going to look at 3 types of lipids. We'll look at the triglycerides or fats, the phospholipids, and the steroids. All right, so triglycerides, also known as fats, their function, they are an energy source. And with their name, tri just refers to the 3 fatty acid chains. And then glyceride just refers to the glycerol molecule. And glycerol is just a modified sugar that is part of the triglyceride. Okay, and over on the right is what a triglyceride molecule looks like. We've got the glycerol here on the top in the tan. So that's the modified sugar. And then attached to that, we've got 3 fatty acid chains. Okay, and then the fatty acid chains that are on the triglycerides can either be saturated fatty acids or unsaturated fatty acids. And what we mean when we say that they're saturated fatty acids, we just mean that they're saturated with hydrogens. So anywhere we can put hydrogens, there are hydrogens. So there's no double bonds between the carbons anywhere. So that just means that they're going to be straight and they can pack together, and so they're going to be solid at room temperature. So a good example of this would be animal fats, butter, those would be saturated fatty acids. The unsaturated fatty acids, we're going to have double bonds between them, and they're not going to be saturated with hydrogens. So we haven't put hydrogens everywhere we could, and so some double bonds form between the carbon atoms. And that allows the fatty acid chains to bend or kink, and so they can't pack together as closely, and so they're going to be liquid at room temperature. So for example, olive oil would be an unsaturated fatty acid. Here's an example of a triglyceride, and we've got 3 different fatty acids attached to the glycerol molecule. So our first fatty acid chain is a saturated fatty acid. So we've got no double bonds anywhere, so it's going to be straight. And then our next fatty acid chain is unsaturated, so you can see there's a double bond there, and so you can see that it's got a bend or a kink in it. And then our last fatty acid chain is also unsaturated, and so you can see that there's 2 double bonds there, and this one would actually be a polyunsaturated fatty acid because you've got multiple double bonds there, and so you can also see that it's bent as well. Our next lipid is the phospholipid. And remember that they're really important because they make up the plasma membrane. And the structure of the phospholipid, remember that they have a hydrophilic head, and then they've got 2 fatty acid chains hanging off that are hydrophobic. So that makes the phospholipid amphipathic. So that's a great word to know. So remember that the term amphipathic just means that one part of the molecule is hydrophilic and the other part is hydrophobic. So because the phospholipids are amphipathic, when they are in water, the hydrophilic heads are going to face out towards the water, and the hydrophobic fatty acid tails are going to point inwards to get away from the water, and so they're going to form a phospholipid bilayer, and that's going to form our cell membrane. All right, and the last lipid we're going to take a look at are steroids. And you can recognize a steroid because they have 4 carbon rings that are fused together. So that will give away a steroid. And steroids will include cholesterol. And cholesterol is used in the plasma membrane of eukaryotic cells, and they will fit in between the layers of the phospholipids, and they help make sure that the membrane maintains the correct fluidity, because you don't want the cell membrane to be too solid, and you also don't want it to be too liquid. So that's the function of the cholesterol in the eukaryotic cell membrane. The prokaryotes also have a steroid in their cell membrane. It's similar to cholesterol, but it's not cholesterol.